Their discoveries have shown us that Earth has always been a place of wonder, danger, and continuous transformation. From the depths of ancient sinkholes to the edges of knowledge itself, we're about to explore the most fascinating discoveries that have reshaped our understanding of life, death, and everything between. This documentary examines some of the most incredible paleontological breakthroughs, mysterious creatures that seem pulled from mythology, the astonishing evolution of dinosaurs into forms we never imagined, and perhaps most remarkably, the microscopic organisms that can survive anything, literally anything, that the universe throws at them. So settle in, find a comfortable place to watch, and let us know in the comments where you're viewing this history video and what time you're watching it. Be sure to subscribe to the channel for future videos exploring the mysteries of our world. Now, let's begin our descent into the deep past. Part 1. The Fossil Record and Ancient Discoveries. The foundation of everything we know about prehistoric life rests upon a single, profound concept: fossils are messages from the past. They are nature's way of leaving behind a record of what once was. But understanding these messages requires patience, expertise, and sometimes, remarkable luck. The Florida's sinkhole, gateway to a forgotten world in the state of Florida, beneath the modern world of highways and cities, lies something extraordinary. Deep underground, natural sinkholes have created perfect preserves for ancient life. These sinkholes form when limestone bedrock dissolves beneath the surface, creating cavities that eventually collapse, leaving behind depression-like formations. But what makes Florida's sinkholes so special is their ability to preserve organic material in ways that normal fossil sites simply cannot. Around 1251 in our timeline of discoveries, paleontologists made remarkable finds within one of these sinkholes. The creatures found here came from the Irvingtonian period, a geological age that has long remained mysterious to science. The Irvingtonian period, spanning roughly from 300,000 to 100,000 years ago, represents a transitional time in Earth's history, a period when the world was shifting, climates were changing, and species were adapting in remarkable ways. The sinkhole acted like a time capsule, perfectly sealed from the outside world. Over thousands of years, animals ventured into these pits and became trapped. Some fell accidentally. Others may have been drawn by the promise of water or shelter. Regardless of how they arrived, their remains settled into the anaerobic environment, an oxygen-free zone where decomposition happens at a glacially slow pace. This lack of oxygen is crucial. Without oxygen, bacteria and fungi cannot efficiently break down organic material. Bones remain intact. Teeth preserve their structure. Even soft tissues can be partially maintained. What paleontologists uncovered in these sinkholes painted a picture of a world teeming with large mammals. There were ground sloths, creatures that seem almost impossibly large and ungainly to modern eyes. These weren't the tiny sloths we see hanging from trees in tropical rainforests today. Ground sloths, known scientifically as Megatherium and related genera, could reach the size of elephants. An adult ground sloth might stand 20 feet tall when rearing up on its hind legs, using its powerful tail for balance. These creatures were herbivores, spending their days browsing on vegetation, stripping leaves and branches from trees using their long, curved claws. Alongside the ground sloths, scientists found the remains of saber-toothed cats, fierce predators with elongated canine teeth that could measure up to 7 inches in length. These weren't the lithe hunters of the African savanna. Saber-toothed cats were stockier, more powerful, built for crushing prey and delivering devastating bites to the throat and belly. They hunted the massive herbivores that shared their world, and occasionally, the sinkholes captured the final moments of both predator and prey. The sinkhole deposits also revealed information about climate and environment. Pollen samples, preserved in the anaerobic mud, told scientists what plants grew in the surrounding landscape. Seeds and plant remains showed what the megafauna were eating. Isotope analysis of bones revealed dietary preferences and migration patterns. Even the spacing of bones sometimes told stories of how animals died. Were they killed here, or did they wander in already weak and dying? But the most important aspect of these discoveries was how they filled a gap in our knowledge. The Irvingtonian period had been poorly studied, partly because it's a transitional zone in geological time. It comes after the Pleistocene megafauna flourished, and before the extinction events that wiped out most large animals at the end of the last ice age. These sinkholes gave scientists a window into how ecosystems functioned during this critical period, how predators and prey coexisted, and how populations were responding to environmental changes. The sinkhole discoveries also demonstrated something crucial about paleontology in the modern era. It's not just about finding bones and displaying them in museums. Modern paleontologists use advanced dating techniques, chemical analysis, genetic sequencing, and computer modeling. Fossils are now subjected to CT scanning, revealing internal structures that would have been impossible to see just decades ago. Isotope ratios in bones and teeth provide information about diet and migration. DNA extraction, though limited for specimens this old, sometimes yields remarkable insights. These discoveries remind us that our planet is layered with history. Every layer of rock represents time. Every fossil represents a life lived. The sinkhole preserved not just the skeletons of creatures, but the context of their lives, the plants they ate, the predators that hunted them, the climate they endured. Understanding the Irvingtonian period to truly appreciate the significance of the sinkhole discoveries, we must understand the Irvingtonian period itself. This age comes roughly 300,000 to 100,000 years ago, placing it squarely in the Quaternary era, the most recent major division of geological time. This was the age of megafauna, a time when the largest land animals the world has ever seen roamed the continents. The Irvingtonian world was not uniform. Different regions experienced different climates. Some areas were verdant grasslands. Others were forested. Some experienced seasonal rainfall patterns similar to modern savannas. The megafauna adapted to these various environments, developing specialized feeding strategies and behaviors. The ground sloths that dominated parts of this world were remarkable in their adaptation. With their long arms, powerful claws, and ability to rear up on their hind legs, they accessed vegetation that other herbivores could not reach. Some scientists believe they fed on trees up to 20 feet high, stripping bark and leaves with systematic efficiency. Their massive size provided advantages in food processing. A larger digestive system could ferment plant material more efficiently, extracting maximum nutrition from tough, fibrous plants. But size comes with challenges. A ground sloth would need to consume enormous quantities of vegetation each day. Paleontologists estimate that an adult megatherium needed perhaps 300 to 400 pounds of plant material daily. This required movement across large territories. The sinkhole deposits suggest that individual animals may have ranged across areas of dozens of square miles, following seasonal vegetation patterns. The saber-toothed cats that hunted these herbivores were equally specialized. Their massive forelimbs were used to grapple with and immobilize prey. The elongated canines delivered a killing bite, likely targeted at the throat to sever blood vessels or damage the windpipe. However, hunting such massive prey was dangerous. Injuries were common. Paleontologists have found saber-toothed specimens with healed fractures in their bones, evidence of past conflicts that didn't end their lives but certainly caused suffering. The predator-prey dynamics of the Irvingtonian period were different from what we see in modern ecosystems. With fewer predator species overall, individual predators may have ranged more widely and sometimes faced little competition for kills. Scavenging was probably more common than active hunting. A large predator could make a kill and feed on it for several days, with smaller scavengers coming afterward to clean up the remains. This feeding cascade meant that a single large herbivore's death could provide sustenance for multiple predator and scavenger species. Climate change was already occurring during the Irvingtonian period. Glaciers were advancing and retreating in northern regions, causing sea levels to fluctuate and coastlines to shift. These changes forced species to adapt, migrate, or go extinct. The sinkhole deposits show evidence of these pressures. Some species that appear in older deposits vanish from younger ones. New species appear as the climate shifts, having migrated from other regions where they previously thrived. The sinkhole environment itself created a unique selective pressure. Certain species were more likely to become trapped than others. Small animals might more easily escape. Large animals with poor swimming ability or low intelligence might fall more readily. This bias means that sinkhole deposits don't always represent the true composition of the surrounding ecosystem. They're biased towards certain species, but they're biased in informative ways. By understanding which animals got trapped, scientists can learn about their behavior, their intelligence, their habitat preferences, and their vulnerability to environmental challenges. The fossil preservation process. How creatures become stone. Before we continue our journey through prehistoric discovery, it's important to understand how fossils form. Preservation is the crucial bridge between life and geological record. Without it, no fossils would exist, and paleontology would be impossible. Fossilization occurs through several different processes, and the method of preservation often tells us something about the environment in which a creature died. The sinkhole deposits demonstrate one particularly effective method: rapid burial combined with anaerobic conditions. When an animal dies in normal circumstances, its body is immediately subject to decomposition. Bacteria and fungi break down soft tissues within days or weeks. Scavengers consume the remains. Weather and water movement scatter the bones. Most of the evidence of that creature's existence disappears within a few years. But occasionally, special circumstances intervene. In the sinkholes, when animals became trapped and died, their bodies sank into the muddy bottom. The mud quickly covered the carcass, and the anaerobic conditions prevented rapid decomposition. The bones, protected from scavengers and weathering, began the process of fossilization. Minerals in the groundwater slowly infiltrated the bone, replacing organic molecules with inorganic ones in a process called permineralization. The bone's original structure was preserved, but its chemistry changed. What had been living bone became stone, so chemically transformed that it was nearly imperishable. But permineralization is just one fossilization pathway. Replacement occurs when the original bone is completely replaced by minerals like silica or pyrite. The result can be such perfect replacement that even cellular-level structures are preserved. Sometimes, replacement preserves original structures so faithfully that a fossil bone is actually harder and more detailed than the original bone ever was. In some cases, fossils form without any of the original material being preserved at all. Molds and casts occur when an organism is buried in sediment. The sediment hardens around the organism, and then the organism dissolves away, leaving a cavity, the mold, that exactly matches the organism's shape. Later, new minerals may fill that cavity, creating a perfect cast of the original form. The sinkhole deposits primarily preserve bones through permineralization and replacement, but sometimes soft tissue impressions survive. Ancient skin, muscle, and even internal organs can leave imprints in the surrounding mud. These imprints are fragile and rare, but when they occur, they provide unparalleled information about what an ancient creature actually looked like. One of the most important implications of understanding fossilization is recognizing that the fossil record is incomplete. The conditions necessary for fossilization are rare. Most creatures that ever lived left no fossil record. The creatures that did fossilize were often those in particular environments: lake bottoms, river systems, volcanic ashfalls, sinkholes, where burial was likely. This means we see a biased view of prehistory. We have excellent records of aquatic and semi-aquatic creatures because they are easily buried in sediment. We have less complete records of creatures from dry uplands and forests, where decay and scavenging were more rapid. The sinkhole deposits, therefore, provide a valuable perspective on a world we'd otherwise barely glimpse. They show us the Irvingtonian megafauna in their habitat, frozen in time by geological accident. Dating the past. How scientists know when things happened. One of the most critical questions in paleontology is not what, but when. How do scientists know that a fossil is 100,000 years old rather than 10,000 years old? Or 1 million years old? Several dating methods exist, and they often confirm each other, providing scientists with confidence in their timescales. The sinkhole deposits were dated using multiple methods, each contributing pieces to the chronological puzzle. Radiocarbon dating is perhaps the most famous method, but it has limitations. This technique measures the decay of carbon-14, a radioactive isotope of carbon that's created in the atmosphere by cosmic rays. Living organisms continuously exchange carbon with the atmosphere, maintaining a consistent ratio of carbon-14 to normal carbon-12. But when an organism dies, it stops exchanging carbon with the atmosphere, and the carbon-14 begins to decay at a known, measurable rate. By measuring how much carbon-14 remains in a sample, scientists can calculate how long ago it died. However, radiocarbon dating is only effective for samples up to about 50,000 years old. Beyond that, so little carbon-14 remains that measurements become unreliable. The Irvingtonian period deposits required other dating methods. Amino acid racemization is another technique useful for older samples. Amino acids, the building blocks of proteins, exist in two forms that are mirror images of each other, left-handed and right-handed versions. Living organisms preferentially use left-handed amino acids. After death, chemical reactions slowly convert left-handed amino acids into right-handed forms in a process called racemization. By measuring the ratio of these two forms, scientists can estimate how long ago the organism died, with effectiveness extending back hundreds of thousands of years. Uranium series dating works on a similar principle but examines the decay of uranium and its daughter products in bones and teeth. Uranium is incorporated into bone and enamel when it forms, and different uranium isotopes decay at known rates. By measuring the ratios of parent and daughter elements, scientists can calculate age. These dating techniques aren't perfect. Each has assumptions and limitations. But when multiple methods on the same sample produce consistent results, confidence in the age determination increases dramatically. The sinkhole deposits produced consistent dates across multiple techniques, confirming the Irvingtonian age assignment. Beyond the Irvingtonian period deposits themselves, the broader geological context provides additional temporal anchors. The rocks surrounding the sinkholes, dated using other methods, confirm the age estimates. Pollen samples, compared to other pollen-dated deposits of known age, provide corroboration. The cumulative effect is a robust chronological framework. This chronological precision is crucial because it allows us to ask sophisticated questions about how ecosystems change through time. Comparing deposits that are 50,000 years apart shows how species composition shifted. Comparing deposits 100,000 years apart reveals major ecological transitions. This temporal resolution transforms fossils from mere curiosities into data points in a grand narrative of environmental and evolutionary change. Connecting the dots. What the sinkhole tells us about the past. The sinkhole deposits from Florida represent more than just bones and rock. They're a window into daily life in a vanished world. By analyzing the bones, teeth, and surrounding sediments, paleontologists can reconstruct aspects of behavior, social structure, and ecology that would otherwise be pure speculation. The bone damage patterns, for instance, reveal hunting and feeding strategies. Bite marks on bones, made by the teeth of predators and scavengers, show which animals fed on which. Tooth marks of saber-toothed cats appear on some prey bones, confirming that these predators actively hunted and consumed the available megafauna. But other bones show tooth marks from smaller carnivores, dire wolves, extinct bears, saber-toothed cats of smaller species. This layering of predation pressure tells us that a killed megaherb wasn't alone in the kill site. Multiple predators scavenged on carcasses in a complex feeding hierarchy. The age structure of the preserved animals provides additional insights. Bones from juvenile animals suggest that breeding occurred locally, that calves were born and raised in the region. Bones from elderly animals with worn teeth and arthritis show that some individuals achieved advanced age despite the dangers of predation and environmental hardship. The ratio of young to old suggests population demographics, which can indicate whether the population was growing or declining. Wear patterns on teeth reveal diet in detail. Different plant types create different wear patterns. Grass-eating herbivores develop distinctive tooth striations. Browsers that feed on leaves and branches show different patterns. By analyzing tooth wear from the sinkhole deposits, scientists determined that the ground sloths were primarily browsers, feeding on leaves and woody material, not grazers. This finding aligns with their morphology. Their long arms and claws are well adapted for reaching vegetation in trees, not for digging in soil to find roots and tubers. The sinkhole deposits also reveal seasonal patterns. Some bone assemblages show evidence of animals dying during particular seasons, perhaps during drought when water became scarce and desperate animals congregated at remaining water sources, like the sinkhole itself. Other assemblages show more random seasonal distribution, suggesting a more stable environment, or a more mobile population that wasn't forced into concentration by environmental stress. Stable isotope analysis adds another dimension of understanding. Oxygen isotopes in bones and teeth reflect the water sources that an animal drank. Different water sources have different isotope ratios, allowing scientists to determine whether an animal migrated seasonally or remained in place. Carbon isotopes reflect diet type at the most fundamental level. Different plant types incorporate carbon differently, creating dietary signatures in bone chemistry. By analyzing these signatures, scientists can determine whether an animal was a pure herbivore, or whether it occasionally consumed different plant types than its primary diet. The sinkhole deposits, viewed as a complete system, show us a functional ecosystem. Herbivores fed on available vegetation. Predators hunted herbivores. Scavengers consumed leftover kills. Plants grew, died, and decomposed, returning nutrients to the soil. Weather and climate fluctuated on various timescales. Birth and death occurred constantly. This wasn't a static, perfectly balanced environment. It was dynamic, stressful, and continuously changing. Most remarkably, it was an ecosystem teetering on the edge of transformation. Within the next 50,000 to 100,000 years, most of these megafauna would be extinct. The Irvingtonian deposits show us the moment before the Great Dying, a glimpse of a world that was already beginning to change, unaware of the extinctions to come. Mysterious creatures in the boundaries of knowledge, the coelacanth, a living fossil returns from the dead sometimes. Science is shocked by nature. Sometimes, decades or even centuries of certainty are overturned in a single moment. The coelacanth represents one of the most dramatic examples of this phenomenon in the history of paleontology. Around 1919, in our exploration of mysterious creatures, we encountered the coelacanth, a fish that shouldn't exist in the modern world. According to all available fossil evidence, coelacanth went extinct approximately 66 million years ago, at the same moment that non-avian dinosaurs disappeared from Earth. These fish had thrived during the Mesozoic era, leaving behind a fossil record spanning hundreds of millions of years. Then, abruptly, they disappeared from the fossil record, and scientists concluded they'd gone extinct along with the dinosaurs. This conclusion stood for nearly 66 million years, unchallenged by fossil evidence. Paleontologists categorized the coelacanth as a Lazarus species, named after the biblical figure Lazarus who was resurrected from death. Lazarus species are organisms that disappear from the fossil record, presumably going extinct, only to reappear later, either as actual living populations or as new fossil discoveries after a long gap. Scientists knew such species existed in theory. The fossil record has gaps and biases. Species could disappear from the record without actually going extinct, living on in areas or environments that didn't preserve fossils well. But the coelacanth wasn't a theoretical curiosity. It was a real, living fish that existed in waters near East Africa and Indonesia. In 1938, fishermen caught a coelacanth in their nets, and the specimen was identified by a museum curator who recognized it as an organism identical to creatures known only from fossils hundreds of millions of years old. The discovery sent shockwaves through the scientific community. How could a fish thought extinct for 66 million years suddenly appear in modern nets? The answer lies partly in understanding habitat and the fossil record's biases. Coelacanth live in deep ocean waters, often at depths of 400 to 700 meters below the surface. At these depths, fossils form poorly. The sediments are fine and easily disturbed. Scavengers are present but sparse. Most dead organisms sink to the very deepest parts of the ocean, where they're consumed by deep-sea creatures or settle into sediments, with almost no other organisms. The fossil record of deep ocean environments is sparse compared to shallow seas, coastal areas, or lake systems. Coelacanth became adapted to deep-sea life over millions of years. Unlike their ancient ancestors that swam in shallow tropical seas, modern coelacanth preferred deep, cool water. This habitat shift meant that as climate changed throughout the Cenozoic era, the time period following the dinosaur extinction, coelacanth retreated to deeper waters where they left fewer fossils. To paleontologists studying shallow water deposits, it appeared they'd gone extinct. In reality, they'd simply moved to an environment where fossilization was less likely. The rediscovery of the coelacanth illustrates a profound truth about extinction. Species don't disappear from Earth just because they disappear from the fossil record. They persist in habitats, sometimes marginal or unusual habitats, sometimes out of sight. The fossil record is a biased sample of the true history of life. This bias means paleontologists must constantly remain humble about their conclusions. A species called extinct might be alive in some corner of the ocean. A species thought to be thriving worldwide might actually be rare and localized. The coelacanth itself became scientifically important for other reasons beyond its survival against the odds. Its anatomy provided a living link to ancient fish, specifically, a glimpse of what transitional forms between fish and tetrapods, the four-legged vertebrates, might have looked like. Coelacanth possess lobed fins, paired appendages that are more limb-like than typical fish fins. These lobed fins are the closest modern analog to the transformed fins that became the legs of early tetrapods. By studying living coelacanth, paleontologists gained insights into how fins became limbs, how aquatic animals transitioned to land, and how evolution had sculpted bodies over hundreds of millions of years. The discovery that coelacanth survived also changed how paleontologists approached extinction. It became clear that absence of evidence isn't evidence of absence. Species that disappeared from one region might persist in another. Species that vanished from the fossil record might be living in habitats not being fossilized. This realization inspired paleontologists to explore unusual environments, deep caves, isolated ocean basins, remote islands, where living fossil species might persist. The coelacanth also became a conservation concern. Here was a species that had survived the extinction event that killed the dinosaurs, had persisted for millions of years in the deep ocean, and now faced extinction from human fishing. A species that had survived 66 million years of natural history was threatened by civilization. This paradox highlighted humanity's impact on nature, and the need for conservation efforts even for species that seemed safely hidden in the deep. The Cambrian period, when life exploded into diversity moving further back in time to approximately 541 million years ago, we encounter the Cambrian period, one of the most significant epochs in Earth's history. At roughly 1801 in our exploration, we focus on this explosion of life that fundamentally transformed Earth's oceans and established many of the major animal body plans that persist to the modern day. The Cambrian period represents a dramatic transition in the fossil record. The rocks older than the Cambrian, stretching back hundreds of millions of years before the Cambrian, show a relatively sparse fossil record. Most organisms were simple, single-celled creatures or soft-bodied animals that left few traces. Then, in what seemed like an evolutionary eye blink, the Cambrian exploded with diversity. Trilobites, which would dominate oceans for the next 270 million years, suddenly appeared in the Cambrian. Arthropods of every description proliferated. Mollusks, echinoderms, and representatives of virtually every modern animal phylum appeared for the first time in the fossil record. It was as if evolution had been waiting in the wings and suddenly took center stage. Scientists have proposed various explanations for this Cambrian explosion. Some suggest it was a true explosion of speciation and body plan innovation, with evolution proceeding at unprecedented rates. Others argue that it reflected changes in the fossil record. Perhaps oxygen levels had risen, allowing larger animals to evolve and survive. Perhaps the development of shells and hard parts made animals more likely to be preserved as fossils. Perhaps the explosion was partly real innovation and partly a preservation artifact. The truth likely involves multiple factors. Oxygen levels were rising during this period, making complex multicellular life more viable. The evolution of eyes in several Cambrian creatures created a new pressure: predation. Once predators could see their prey, an arms race began. Prey evolved shells and defenses. Predators evolved tools and strategies to penetrate those defenses. This pressure favored the rapid evolution of new body plans and defensive structures. Additionally, the Cambrian period saw the colonization of new ecological niches. Without competition, organisms could diversify rapidly. A simple body plan, a worm-like body with appendages, was modified into thousands of variations, each specialized for a particular niche, feeding on different foods, living in different depths, moving in different ways. This diversification happened over relatively few millions of years, which is why paleontologists call it an explosion. The Cambrian period represents a time of experimentation in body design. Evolution tried forms that later became extinct, designs that didn't prove viable in the long term. But it also established successful designs that would persist for hundreds of millions of years. The body plans established during the Cambrian essentially define animal diversity to this day. Most modern animals are variations on Cambrian themes. The three-eyed Mussura. When evolution experiments with vision around 54 to 31 in our exploration, we encounter one of the Cambrian's most fascinating creatures, the three-eyed Mussura. This organism represents evolution's approach to solving the problem of vision, with an unusual solution that maximizes information gathering about the environment. Mussura was a small organism, measuring mere centimeters in length. Its body was covered with spines and protrusions. But most notably, its head bore not one or two eyes, but three. Two large compound eyes provided lateral vision, giving the creature sight to the left and right. A third eye, positioned more centrally on the head, probably provided upward-facing vision, allowing the creature to detect light and shadow from above. The three-eyed design suggests a creature that needed to monitor its environment from multiple directions simultaneously. Perhaps Mussura was a predator, using its eyes to track prey in the turbid Cambrian waters. Perhaps it was prey itself, needing to watch for danger from multiple angles. Most likely it was both, hunting smaller creatures while avoiding larger ones. The existence of Mussura and other three-eyed Cambrian creatures demonstrates that evolution explores multiple solutions to the same problem. Vision provides obvious survival advantages. Organisms that can see are better hunters, better at avoiding predators, and better at navigating their environment. So it's unsurprising that eyes evolved in the Cambrian. But why did some organisms settle on 2 eyes while others developed 3? Why not 4 or 5? The answer likely relates to evolutionary trade-offs. Eyes are metabolically expensive, requiring significant energy to construct and maintain. They're also heavy and require neural tissue to process the information they gather. There's a cost to having too many eyes. Mussura's 3-eye system represented, perhaps, an optimal balance for a small predatory creature in the Cambrian seas. The 3-eyed Mussura also reminds us that the Cambrian was not a time of fully modern-looking creatures. These weren't miniature versions of modern fish or crustaceans. They were fundamentally different in design, with body plans that seem alien to modern eyes. Yet they were successful. They reproduced, competed, evolved, and left fossils behind. Their lineages either persisted or went extinct, but their influence on subsequent evolution was profound. The mysterious Tully Monster, evolution's enigma. Perhaps no creature from the Cambrian has proven more puzzling than the Tully Monster, which appears at 31:44 in our journey. This organism has frustrated paleontologists for decades, never fitting neatly into any established animal group. It remains, in many ways, a mystery. The Tully Monster was a small creature, usually less than 4 inches long, with a body plan that seems almost designed to confuse classification. It had a long, slender body with a distinctive trunk or snout protruding from its head. It had fins or fin-like structures along its body. Most peculiarly, it had what appeared to be eyes set on a bar or projection that extended to the sides of its head, an arrangement unlike virtually any other known creature. For decades, paleontologists debated what the Tully Monster was. Was it a primitive fish? A mollusk? An arthropod? A unique creature with no close modern relatives? The problem was that it had characteristics of several different animal groups, but didn't fit neatly into any of them. Detailed fossil specimens, preserved in fine-grained shales, eventually revealed more information. The creature had gill slits, suggesting it was a chordate, the group that includes all vertebrates, along with some invertebrates. It had a distinct head, trunk, and tail. The stubby appendages might have been primitive fins or might have served some other sensory or feeding function. The trunk was curved in a way that suggested it could contract and expand, perhaps for feeding or movement. The most likely interpretation, based on the best available evidence, is that the Tully monster was an early chordate, a primitive relative of modern fish. Its unique characteristics suggest that it represents a distinct evolutionary lineage that left few descendants. It thrived for a time, then disappeared from the fossil record, leaving behind only its puzzling remains to tease modern paleontologists. The Tully monster serves as a powerful reminder of the diversity that existed in ancient oceans. Not all evolutionary lineages were successful in the long term. Some left only a few fossil specimens before vanishing completely. The Tully monster represents evolution experimenting with a unique body plan that, ultimately, proved unsuccessful. But for the millions of years it persisted, it was a successful organism, reproducing, hunting, and surviving in the competitive Cambrian seas. The continued mystery of the Tully monster also highlights the limitations of our knowledge. Despite all our paleontological techniques and scientific advances, seem cryptic. Their relationships to other organisms aren't entirely clear. Their ecology and behavior must be inferred from fragmentary evidence. The Tully monster reminds us that nature is more complex and creative than we've yet fully understood. Why the Cambrian matters. The origin of animal diversity. The Cambrian period was the crucible in which modern animal diversity was forged. Without the Cambrian, Earth today would be a very different place. If that evolutionary explosion hadn't occurred, if those body plans hadn't been established and perfected, the subsequent history of life would have unfolded entirely differently. The Cambrian demonstrates that evolution is not a gradual, smooth process, though it can be that. Evolution is also capable of rapid diversification when conditions permit. When new ecological niches open up, either because of environmental change or the evolution of a new innovation like the eye, speciation and morphological change can accelerate dramatically. The Cambrian shows evolution in fast-forward, trying out thousands of designs in a few million years. The period also demonstrates the importance of hard parts in the fossil record. When organisms evolved shells, spines, and exoskeletons, they became much more likely to be preserved as fossils. This is partly because hard parts are harder to decompose, and partly because the presence of hard parts often indicates a shift in ecology or lifestyle that makes fossilization more likely. The Cambrian radiation of hard-bodied organisms is reflected in a dramatic increase in fossil preservation and diversity. The creatures of the Cambrian were not primitive in the sense of being simple or ineffectual. They were sophisticated, specialized, and perfectly adapted to their niches. The term, primitive, often carries a connotation of inferiority, but Cambrian creatures were not inferior. They were different. They represented different evolutionary solutions to the problems of surviving and reproducing in ancient oceans. Many of those solutions were quite elegant. The strange dinosaurs, evolution gone wild, from the tyrant lizards to the bizarre theropods. When people think of dinosaurs, they often picture the classic forms: Tyrannosaurus rex, the apex predator with massive teeth and tiny arms; Triceratops, the heavily armored herbivore; Stegosaurus with its distinctive back plates. These creatures were real and successful, but they tell only part of the dinosaur story. The true diversity of dinosaurs, and the bizarre forms some achieved, is far more remarkable than popular imagination usually encompasses. Dinosaurs dominated terrestrial ecosystems for approximately 165 million years, from their first appearance in the Middle Triassic period through their extinction at the end of the Cretaceous period. That's a longer span of dominance than mammals have enjoyed, despite mammals being common now. In that 165 million years, dinosaurs evolved into thousands of species, filling virtually every from the depths of ancient sinkholes to the edges, but doesn't eliminate it. The genetic novelty of extremophiles. Studying the genomes of extremophiles has revealed another remarkable feature. They possess genes unlike those found in any other organisms. These genes encode proteins that perform functions never seen before in non-extremophile organisms. Some of these novel genes are related to DNA repair, allowing extremophiles to survive radiation damage that would be lethal to other organisms. The DNA repair genes of extremophiles are more efficient, more robust, more capable than those of non-extremophiles. Other novel genes encode proteins that stabilize cellular structures under extreme conditions. Some of these proteins are heat-stable to degrees seen nowhere else in the biological world. Others stabilize membranes in the absence of water. Still others protect proteins from damage by extreme conditions. The existence of these novel genes and proteins suggests that extremophiles have evolved unique solutions to survival challenges. These solutions are not just variations on standard biological themes. They're innovations, novel approaches to solving problems. This novelty makes extremophiles valuable for biotechnology. Scientists have successfully transferred extremophile genes into other organisms, attempting to transfer resilience across species boundaries. A thermophilic enzyme from heat-loving bacteria has been incorporated into cold-loving microorganisms. Protective proteins from tardigrades have been studied for their potential to stabilize vaccines and biologics during storage. The DNA repair genes of extremophiles might eventually be used to protect human cells from radiation damage. The philosophical implications of extremophiles. The existence and study of extremophiles raises profound philosophical questions about the nature of life itself. What does it mean to be alive? Is metabolism essential, or can life exist in a state of complete metabolic suspension? Is movement essential, or can a stationary organism be alive? Is sensory perception essential? Extremophiles challenge all these assumptions. A desiccated tardigrade is arguably alive, yet it exhibits none of the characteristics we typically associate with life. It has no metabolism. It doesn't move. It doesn't sense its environment or respond to stimuli. Yet it's not dead, its DNA intact, its proteins preserved, its potential for life undiminished. This raises questions about how we define death. Is a desiccated organism dead? Is a frozen organism dead? Or is death a process from which there is no return, and as long as an organism can be revived, it was never truly dead? These are not just semantic questions. They have implications for medicine. Cryogenic preservation of human embryos or organs depends on the idea that organisms can survive freezing and be revived later. Understanding how tardigrades and rotifers survive freezing might lead to better methods of preserving human tissues for transplantation. Extremophiles also challenge our assumptions about what is necessary for life. If life can exist without liquid water, in boiling hot springs or frozen glaciers, then the definition of habitable zones around other stars should be reconsidered. Planets we thought uninhabitable might harbor life in forms we haven't anticipated. The cryptids and the wild world beyond knowledge, the Mokombembe, searching for dinosaurs in Africa at 37 to 44 in our exploration, we encounter a creature of legend and myth, the Mokombembe, a supposed dinosaur living in the remote rainforests of Central Africa. The name, in local African languages, translates roughly to one who stops the flow of rivers, and refers to what locals describe as a massive, long-necked creature living in remote rivers and lakes of the Congo Basin. The Mokombembe is a cryptid, a creature whose existence is suggested by folklore and anecdotal reports but lacks concrete scientific evidence. Accounts describe a creature resembling a sauropod dinosaur, long-necked, massive-bodied, semi-aquatic. Some reports suggest it's over 35 feet in length. Locals in the Congo Basin speak of it with a mixture of fear and respect, treating it as a real part of their world. From a scientific perspective, the existence of a living dinosaur in Central Africa is virtually impossible. Dinosaurs went extinct 66 million years ago, excepting birds, their avian descendants. There's no plausible mechanism by which a non-avian dinosaur could have survived for 66 million years hidden in rainforests. Yet the Mokombembe persists in legend, and enthusiasts continue to search for it. Why would legends of a dinosaur-like creature exist in Africa? One possibility is misidentification. Large aquatic animals like Nile crocodiles, hippopotamuses, or unusual fish might be misidentified as dinosaurs by people unfamiliar with these creatures. Another possibility is that the legend preserves a memory of actual megafauna that lived in the region until recently in geological time. Megafauna did live in Africa during the Pleistocene epoch, so it's possible that some of these animals persisted longer in isolated areas than elsewhere. Another possibility is that the legend is purely mythological, with no basis in recent observation. Cultures worldwide have created myths and legends about exotic creatures. These legends may have been inspired by fossil discoveries, ancient people finding dinosaur bones and constructing stories around them. Or they may have been pure fantasy, cultural creations rather than reports of real animals. Scientific expeditions to search for the Mokombembe have found no concrete evidence of its existence. No photographs, no video, no physical specimens. These expeditions, however, have discovered new species previously unknown to science. The rainforests of Central Africa are so vast and unexplored that discovering animals unknown to science is not unreasonable. But those discoveries have been of reasonable animals, new species of fish, reptiles, and mammals, not living dinosaurs. The Gobi Desert Death Worm. When legend meets science at 41:58, we encounter another cryptid, the Gobi Desert Death Worm, a creature of Mongolian legend that supposedly lurks beneath the sands of the Gobi Desert. Local stories describe a creature called the albacorcoy, a long, worm-like animal that moves through the sand like a fish through water, that can spray corrosive venom, and that can kill with a single touch. The Gobi Desert Death Worm became popular in Western culture in the 1920s through explorer and paleontologist Roy Chapman Andrews. Andrews led expeditions into the Gobi Desert seeking dinosaur fossils. While there, he heard local legends of the death worm. Upon returning to the West, he published accounts of the creature, sparking decades of fascination and speculation. What is the Gobi Desert Death Worm? Several possibilities exist. One is that it's purely mythological, a cultural creation of people living in an environment they found frightening and mysterious. Another is that it represents a myth of a real animal, a large lizard or snake, perhaps, or an unusual burrowing mammal. Another possibility is that it represents a memory of an animal that actually lived in the Gobi but has since gone extinct or migrated elsewhere. Actually, modern paleontologists searching the Gobi have found evidence that at least one species fitting the death worm description might have plausible ancestors. Ancient burrow-dwelling creatures, some of which were worm-like and potentially venomous, lived in environments similar to the modern Gobi. Over time, as the Gobi became hotter and drier, these creatures may have gone extinct, leaving behind only legends. More recent scientific expeditions to search for the Gobi Desert Death Worm have found no evidence of its existence, as described in legend. No specimens, no photographs, no clear physical evidence. Yet the expedition teams have gathered comprehensive data about the Gobi's fauna and environment, making contributions to science despite failing to find the legendary creature. The role of myth in understanding ancient life. The searches for Mokombembe and the Gobi Desert Death Worm, while ultimately unsuccessful in finding the cryptids themselves, illustrates something important about how humans relate to nature and history. We're drawn to mysteries. We create narratives to explain phenomena we don't fully understand. We have always done this. Ancient peoples, discovering fossil bones and unable to place them in any framework of known animals, created stories. A giant bone became evidence of a giant creature. A fossil shell on a mountain became proof that the sea had once covered high ground. Myths and legends preserved kernels of observation and experience, wrapped in interpretive frameworks that made sense given the knowledge available at the time. These myths are not wholly false, nor wholly true. They're cultural expressions of genuine human experiences and observations, filtered through the lens of available knowledge. The fossil discoveries that likely inspired myths about giant creatures were real. The observation that seashells are found in mountains is real. We now know that tectonic processes raise ancient seafloor into mountains. But the interpretive frameworks, stories of giants, of catastrophic floods, were mythological attempts to make sense of these observations. Modern paleontology and geology have provided better frameworks, more accurate models of past events and creatures. Yet we still encounter the same human tendency to create stories, to be fascinated by mysteries, to wonder about unknown creatures and vanished worlds. Cryptozoology, the study of cryptids, is a modern manifestation of this ancient human impulse. The value of cryptozoology is not necessarily that cryptids exist, but that cryptozoologists often make scientific observations in their searches. They collect data about real animals, about ecology, about environmental conditions. Even unsuccessful searches for cryptids contribute to scientific knowledge about the regions being searched. Cryptids as windows into human cognition. The persistence of belief in cryptids despite lack of evidence says something important about human psychology and cognition. We're pattern recognition creatures. We see patterns in data, even when the patterns aren't really there, a phenomenon called apophenia. We create narratives to make sense of observations. We assign agency and intention to natural phenomena. Cryptids fill a role in human psychology. They represent the unknown, the possibility of discovery, the reminder that the world is larger and stranger than our everyday experience suggests. They appeal to and our desire to find. This believers in cryptids are foolish. Its terrains have innates that predispose unexplained phenomena when those consistent with cultural narratives and legends. Science doesn't eliminate this human tendency toward creating narratives about the unknown. Rather, science provides alternative narratives, grounded in evidence and testable hypotheses. The narrative science provides may be less emotionally resonant than the legend of a dinosaur living in remote rainforests, but it's more reliable as a guide to understanding reality. The persistence of cryptid legends also reminds us of how much of the natural world remains unexplored and unknown. New species of animals are discovered regularly. Ecosystems we thought we understood are revealed to be more complex than we realized. There's genuinely much that we don't know about the living world. The difference between cryptozoology and paleontology is that paleontology uses systematic methods to explore the unknown, while cryptozoology often relies on anecdote and legend. Conclusion 2 Part 1 We have journeyed from the depths of Florida sinkholes, through ancient oceans filled with exotic creatures, into the age of dinosaurs and their bizarre forms, and into the microscopic realm of creatures that defy the limits of biology. We have explored both the solid ground of paleontological science and the speculative realm of cryptid legends. What emerges from this journey is a vision of life, both past and present, as remarkably resilient, remarkably creative, and remarkably diverse. Life has existed on Earth for approximately 3.8 billion years. In that time, it has evolved from simple, single-celled organisms to an incredible array of forms. It has learned to live in every conceivable environment, from the deepest ocean trenches to the frozen poles, from boiling hot springs to the vacuum of space. Each creature we've encountered, the Irvingtonian megafauna, the Coelacanth, the bizarre Cambrian organisms, the strange dinosaurs, the extremophile microorganisms, tells us something about the possibilities of life. Adaptation, evolution, and survival are ongoing processes. The same processes that created the diversity of prehistoric life continue today, shaping evolution in real time. Equally important is what these discoveries tell us about the nature of science itself. Science is a process of asking questions and attempting to answer them through systematic observation and experimentation. When the Coelacanth was rediscovered, decades of certainty about its extinction were overturned. When more complete Deinocheirus fossils were found, previous speculation was proved wrong. This is not a weakness of science, but a strength. Science is self-correcting, continually refining its understanding as new evidence emerges. Paleontology is perhaps uniquely humbling in this regard. We are attempting to understand the distant past, based on incomplete evidence preserved by chance. We must remain constantly aware of the limitations of our knowledge, of the biases in the fossil record, of the gaps in our understanding. Yet despite these limitations, paleontology reveals a past more wondrous and complex than human imagination could create. The study of ancient life also reminds us of our place in Earth's history. Humans are recent arrivals on this planet. Our species has existed for less than 1 million years, a mere eye blink in geological time. We live in the aftermath of extinctions that eliminated previously dominant forms of life. The dinosaurs, the megafauna, the countless species that have gone extinct in Earth's history, their absence is as important a part of Earth's environment as the presence of species that survived. This recognition should inspire both humility and responsibility. Humility, because we are not the culmination of evolution, not the final form toward which all previous life was building. We are simply the current dominant large animal on Earth, occupying a particular ecological role at a particular moment in time. Responsibility, because we have the unique ability to understand and potentially change our environmental impacts. Other species have driven environmental changes. The Great Oxidation event caused by photosynthetic bacteria changed Earth's atmosphere forever. But humans are the first species to understand the potential consequences of our actions and to have the ability to choose a different path. As we conclude part 1 of this documentary, we have set the foundation. We have surveyed the incredible diversity of life that preceded us, the mechanisms by which we know about that life, and the remarkable resilience and creativity that life exhibits. In part 2, we will delve deeper into specific topics, exploring in greater detail the mechanisms of evolution, the ways that life adapts to environmental change, and what the study of paleontology and astrobiology tells us about the future of life on Earth and the possibility of life elsewhere in the universe. Thank you for joining us on this journey through time. We hope you've found the discoveries as fascinating as we have. In the comments, let us know what aspect of prehistoric life fascinates you most. Was it the Cambrian explosion? The strange forms of dinosaurs? The incredible resilience of extremophiles? Your comments help us create content that resonates with viewers and reflects your interests. Subscribe to the channel for part 2, where we'll continue exploring the mysteries of life, the mechanisms of evolution, and the frontiers of paleontological science. The story of life on Earth is far from over. There are still discoveries to be made, mysteries to be solved, and wonders to be uncovered. Until then, keep looking up and down. The earth beneath our feet holds the secrets of our past and clues to our future. Welcome back to our continued exploration of Earth's incredible past and the resilience of life in forms that challenge our understanding of biology itself. If you're just joining us, we recommend watching part 1 first, where we explored the Irvingtonian megafauna, the mysterious coelacanth, the bizarre creatures of the Cambrian period, and the strange dinosaurs that walked ancient Earth. In part 2, we deepen our investigation. We move beyond cataloging the discoveries to understanding the mechanisms that drive them. How does evolution actually work? What causes mass extinctions? How do species adapt to environmental change? What does the study of life on Earth tell us about the possibility of life elsewhere in the universe? These are the questions that animate modern paleontology and astrobiology. We'll explore the practical ways that scientists extract information from fossils, the cutting-edge technologies that have revolutionized paleontological research, and the debates that continue within the scientific community about interpretation and meaning. We'll examine environmental catastrophes that have reshaped life on Earth, the mechanisms of adaptation and evolution, and the fascinating field of astrobiology, the search for life beyond our planet. This is science in action, not as a static body of knowledge, but as an ongoing process of discovery and refinement. Let's continue our journey. Part 2, Mechanisms, Methods, and Mysteries, the Modern Paleontological Toolkit. How scientists read the fossil record. Paleontology in the modern era bears little resemblance to paleontology a century ago. Where past paleontologists relied primarily on visual examination and mechanical excavation, modern paleontologists employ a diverse arsenal of technological and analytical tools that would seem like science fiction to their predecessors. CT scanning and virtual paleontology. One of the most revolutionary technologies in modern paleontology is computed tomography scanning, CT scanning. This technique, borrowed from medical imaging, uses X-rays to create detailed three-dimensional maps of the internal structure of fossils without destroying them. Consider a fossil skull. In the past, paleontologists could only examine the external surface. To see internal structures, they had to break the specimen open, destructive analysis that permanently damaged irreplaceable artifacts. With CT scanning, scientists can examine every millimeter of the skull's interior without a single scratch on the specimen. A CT scan creates hundreds or thousands of thin cross-sectional images. These images are then assembled by computer into a complete three-dimensional model. Paleontologists can manipulate this model, viewing it from any angle, zooming in on specific structures, even digitally, removing, overlying material to see hidden features more clearly. The applications are profound. Deinocheirus' skull was CT scanned. Researchers could examine the internal structure of the brain case, revealing the shape and size of the brain. Knowing brain size and shape provides clues about sensory abilities and intelligence. Large olfactory centers suggest good smell. Large visual centers suggest reliance on vision. The shape and size of different brain regions can be compared to modern animals, suggesting which sensory systems the dinosaur relied upon most heavily. CT scanning has also revealed pathological conditions that wouldn't be apparent from external examination. Cancers, infections, injuries, all leave marks on bone that CT scanning can detect. Paleontologists have found evidence of bone infections in dinosaurs, of arthritis in ancient mammals, of healed fractures in creatures millions of years old. These medical conditions tell us about the quality of life experienced by prehistoric creatures, the injuries they suffered, the diseases they endured. Virtual paleontology extends CT scanning technology even further. Once a fossil is scanned, digital models can be printed using 3D printing technology. Museums can now display replicas of fossils while protecting originals from wear and damage. Scientists can send digital models to colleagues anywhere in the world, eliminating the need for expensive and risky transport of fragile specimens. Models can be manipulated, measured, and analyzed in ways the original would not permit. Virtual paleontology has also enabled new forms of analysis. Digital models of extinct creatures can be subjected to biomechanical simulations. How fast could a particular dinosaur run? What forces would act on its bones during locomotion? Computer models can simulate muscle contraction and movement, providing insights into behavior that would be impossible to obtain from static fossils. Chemical analysis and isotope ratios beyond visual examination. Modern paleontologists employ sophisticated chemical analysis of fossil material. One particularly powerful technique involves analyzing stable isotopes, variant forms of elements that differ slightly in mass but are chemically identical. When organisms incorporate elements into their bodies, carbon into proteins and structural materials, oxygen into water and minerals, nitrogen into proteins, they do so with slight preferences for lighter or heavier isotopes. This preference, called isotopic fractionation, depends on environmental conditions. Organisms living in different climates, at different latitudes, or eating different diets will have different isotopic ratios in their tissues. When fossils are analyzed for their isotopic composition, that composition preserves a chemical record of the organism's environment and diet. Carbon isotopes reveal what plants and animals ate. Plants using different photosynthetic pathways have different carbon isotope ratios, and these ratios are incorporated into herbivores that eat them, and then into carnivores that eat herbivores. Oxygen isotopes are similarly informative. The oxygen isotope ratio in bone and tooth enamel reflects the water the organism drank. Water's oxygen isotope ratio varies with latitude and climate. Cold polar water has a different isotope ratio than warm tropical water. Rainfall in wet seasons has a different ratio than in dry seasons. By analyzing oxygen isotopes, paleontologists can infer where an organism lived and if they have multiple samples from different times in the organism's life, whether it migrated seasonally. Strontium isotopes provide yet another dimension of information. Like oxygen, strontium isotope ratios vary geographically. Different geological regions have characteristic strontium isotope ratios. By comparing the strontium isotope ratio in an organism's teeth to the strontium ratio of the local geology, scientists can determine whether the organism grew up where it died or migrated in from elsewhere. These chemical techniques have revolutionized understanding of prehistoric life. We now know that some dinosaurs migrated seasonally, following food sources and water. We know that some ancient humans migrated across continents. We know the dietary patterns of extinct species, the climates they lived in, the resources they exploited. Genetic analysis. When ancient DNA speaks, perhaps the most dramatic recent advance in paleontology is the extraction and analysis of ancient DNA. Though often impossible with very old specimens, DNA degrades over millions of years. Ancient DNA can sometimes be recovered from specimens that are tens of thousands of years old, and occasionally from even older samples. The process is technically challenging. DNA in ancient specimens is fragmented into tiny pieces, often damaged by chemical processes and contaminated with modern DNA from handling. But modern sequencing technology can handle these challenges. Scientists have developed techniques to identify authentic ancient DNA, to distinguish it from contamination, and to sequence billions of DNA fragments simultaneously. The results have been astounding. We have recovered DNA from Neanderthals, our extinct human cousins, allowing us to understand their genetic differences from modern humans, to trace their evolutionary history, and to determine that interbreeding between Neanderthals and modern humans occurred, leaving genetic traces in modern human genomes. We have recovered DNA from mammoths, extinct relatives of modern elephants. This DNA has revealed genetic changes that occurred after mammoths split from their elephant ancestors, showing how their genomes adapted to cold climates. Interestingly, some mammoth genes show signs of natural selection. Certain mutations became more common over time, suggesting they provided advantages in the icy environments mammoths inhabited. We have recovered DNA from extinct moas, enormous flightless birds from New Zealand. The DNA revealed their evolutionary relationships, showing that moas were more closely related to kiwis than to emus or ostriches, despite superficial similarities. The DNA also revealed that some species of moa had undergone recent genetic changes, suggesting that they were still evolving when humans arrived in New Zealand and subsequently hunted them to extinction. The recovery of ancient DNA has also enabled one of the most ambitious projects in modern biology: de-extinction. Scientists have been considering whether it might be possible to recreate extinct species by using ancient DNA. The most famous case is the woolly mammoth. Some scientists believe that by editing the genome of an Asian elephant to incorporate mammoth DNA, creating a mammophant, they could recreate a creature similar to the extinct mammoth. De-extinction raises philosophical and practical questions. Would a recreated mammoth be truly a mammoth, or merely a living sculpture, a close approximation of the real thing? Would there be ecological value in reintroducing mammoths to replace species lost in the Pleistocene extinctions? Would resources spent on de-extinction be better invested in protecting currently endangered species? These questions remain debated. But the technological capability is advancing rapidly. Within decades, de-extinction of some megafauna species might become reality. This would represent an unprecedented intervention in natural processes, undoing extinction through technology, bringing back a species that has been dead for thousands of years. Biomechanical modeling and behavioral reconstruction. Beyond these analytical techniques, modern paleontologists use biomechanical modeling to understand how extinct creatures moved and behaved. Once a fossil skeleton is digitized, computer models can be created that simulate the skeleton with muscles, joints, and gravity. Consider a question paleontologists have long debated: how fast could Tyrannosaurus rex run? Visual inspection of a skeleton tells us that T. rex had powerful hind legs and a heavy tail. But the actual forces involved in running are complex. Biomechanical models can simulate muscle contraction, joint angles, and movement, calculating the maximum speed achievable without impossible forces acting on the skeleton. Such models have revealed that T. rex was probably not an extremely fast runner, certainly faster than a human, but not as fast as smaller predators like wolves or lions. This finding has changed ideas about T. rex hunting strategy. Rather than being a pursued predator that chased prey at high speed, T-Rex might have been an ambush hunter, taking prey by surprise, or a scavenger that fed on carcasses killed by other predators. Biomechanical models have also been applied to aquatic creatures. Swimming speeds of ancient marine reptiles, flying abilities of pterosaurs, climbing abilities of various dinosaurs, all can be estimated through biomechanical modeling. These estimates are not perfectly accurate. Living organisms are more complex than computer models, but they provide reasonable estimates of what was physically possible. Biomechanical modeling has also been used to test ideas about sexual displays and combat. Some dinosaurs, like Triceratops, had elaborate horns and frills. Were these primarily for defense against predators, or were they sexual displays used in combat between males competing for mates? Biomechanical models can simulate impacts and forces, revealing which structures would be effective in combat, and which would primarily serve as displays. The taphonomy. Understanding how fossils form a crucial aspect of modern paleontology is taphonomy, the study of how organisms become fossils. Taphonomy asks questions about burial, preservation, diagenesis, the chemical changes that occur during fossilization, and all the processes that affect a dead organism between death and its discovery as a fossil millions of years later. Understanding taphonomy is essential because it helps paleontologists interpret fossils accurately. A bone assemblage might look like evidence of a predator's kill site, but taphonomic analysis might reveal that the bones were transported by flowing water from several different locations, creating a false impression of association. Taphonomic analysis includes studying scavenging patterns, trace marks left by various organisms, evidence of chemical alteration, and the spatial distribution of bones. By understanding how various taphonomic processes affect bones, paleontologists can reconstruct what happened to an organism after death and before fossilization. This analysis has overturned some long-held beliefs. For example, bone beds where hundreds or thousands of individuals of the same species are found were once interpreted as evidence of catastrophic mass deaths or hunting by early humans. Taphonomic analysis has shown that many such accumulations form through slower processes, natural deaths over time, with bones accumulating in river channels or lake bottoms where they become buried together. The final fossil assemblage looks like a catastrophe but represents normal mortality spread over thousands of years. Mass extinctions. When life nearly ends, the Big Five, Earth's greatest catastrophes throughout Earth's history, life has survived numerous environmental challenges. Some of these challenges have been devastating, eliminating the majority of species in existence. These major extinction events are called mass extinctions, and 5 are particularly significant, the Big Five extinction events. The first mass extinction occurred at the end of the Ordovician period, approximately 443 million years ago. Glaciation caused sea levels to drop dramatically, eliminating the shallow marine environments where most life existed at that time. Approximately 85% of all marine species went extinct. This event reshaped marine ecosystems fundamentally. The second major extinction occurred at the end of the Devonian period, approximately 375 million years ago. This extinction was probably caused by multiple factors: climate change, ocean acidification, changes in atmospheric oxygen levels, and asteroid impact. Approximately 75% of all species went extinct, with marine life hit particularly hard. The third mass extinction occurred at the end of the Permian period, approximately 252 million years ago. This is the most severe extinction event in Earth's history, with approximately 96% of marine species and 70% of terrestrial vertebrate species going extinct. The cause was probably related to massive volcanic eruptions that released vast quantities of greenhouse gases, causing runaway climate change, ocean acidification, and widespread anoxia, lack of oxygen, in marine environments. The fourth mass extinction occurred at the end of the Triassic period, approximately 201 million years ago. The cause was again probably volcanic activity combined with climate change. Approximately 76% of all species went extinct. The fifth and most recent major extinction occurred at the end of the Cretaceous period, approximately 66 million years ago. This is the extinction event most famous in popular culture, the event that killed the non-avian dinosaurs. The primary cause was impact by a large asteroid, with secondary effects including volcanic activity, climate disruption, and ecosystem collapse. Each of these extinctions followed a similar pattern. Some environmental change occurred, species that could not adapt to the new conditions went extinct, and the survivors radiated into the empty ecological space left behind. Life rebounded after each extinction, but the composition of life on Earth was fundamentally altered. The KPG extinction, the dinosaur extinction, the extinction event 66 million years ago, the Cretaceous Paleogene, KPG, extinction, formerly called the KT extinction, is the most thoroughly studied mass extinction. The reason is its timing and the fossil record. It's recent enough in geological time that the fossil record is comparatively well preserved, and it's old enough that we can see the long-term consequences. The KPG extinction was caused by impact of an asteroid approximately 10 kilometers in diameter in the Yucatan Peninsula of what is now Mexico. The impact created a crater approximately 180 kilometers in diameter, the Chicxulub crater. The impact released energy equivalent to billions of nuclear weapons, creating a fireball visible across the planet, triggering earthquakes and tsunamis, and ejecting massive quantities of dust into the atmosphere. The atmospheric dust was the critical factor. For months or years after the impact, dust and soot from wildfires filled the atmosphere, blocking sunlight. Photosynthesis nearly ceased. Plants died. Herbivores lacking food starved. Carnivores lacking prey starved. Temperatures plummeted as sunlight was blocked. It was a cascade of environmental change that favored only the smallest, most generalist organisms. This scenario explains the extinction pattern. Large animals, with high metabolic demands and lower reproductive rates, couldn't survive the disruption. Small animals, with lower metabolic needs and faster reproductive rates, could survive on limited resources and rebound quickly when conditions improved. Non-avian dinosaurs, which had dominated terrestrial ecosystems for 165 million years, were eliminated entirely. But birds, which are technically avian dinosaurs, living dinosaurs, survived. The smallest theropods, including the ancestors of modern birds, possessed adaptations that allowed survival. Possibly feathers for insulation, possibly small body size allowing survival on limited food, possibly efficient metabolism. Marine ecosystems were equally devastated. Ammonites, which had flourished for 300 million years, went extinct. Mosasaurs, the apex marine predators, disappeared. Marine reptiles and most fish species went extinct. Only small fish and specialized organisms survived. But within a few million years, life rebounded. Mammals, which had been small, insignificant creatures in the shadow of dinosaurs, began to diversify. Birds proliferated, eventually filling the ecological roles dinosaurs had occupied. Fish evolved into new forms. Plants colonized the stripped landscape. Within 10 million years, ecosystems were functioning again, though fundamentally altered. Without dinosaurs, the world became a place where mammals could thrive, eventually leading to the evolution of primates and humans. The KPG extinction, catastrophic as it was, set the stage for the rise of mammals and the eventual emergence of humanity. Extinctions and ecosystem recovery. What's remarkable about mass extinctions is not just that they happen, but that life recovers. Each of the big 5 extinctions was followed by what's called an adaptive radiation, an explosion of speciation as surviving species evolved into new forms, filling empty ecological niches. The recovery patterns tell us something important about how evolution works. Evolution is not directed toward an ultimate goal. It doesn't produce better species. Rather, it produces species adapted to available niches. When many niches become vacant due to extinction, many opportunities for evolution arise. Species that were previously marginal or unsuccessful suddenly have advantages. Speciation accelerates. Morphological change accelerates. Within a few million years, new species have evolved to fill most of the empty niches. This pattern has occurred repeatedly in Earth's history. After the Permian extinction, which was the most severe, it took perhaps 10 million years for marine ecosystems to recover and 30 million years for terrestrial ecosystems to fully restore diversity. After the KPG extinction, mammals diversified rapidly in the Paleocene and Eocene epochs, evolving into the major groups we see today within just 20 million years. The rapidity of recovery suggests that the genetic variation needed for rapid evolution exists in surviving populations. When natural selection pressures change, species can evolve quickly to exploit new opportunities. This same principle operates in ecological time. When food sources change, animals adjust their diet within a generation. Evolution works on longer timescales, but the potential for rapid change is present. Understanding extinction risk in the modern world. The study of mass extinctions has direct relevance to understanding modern extinction risks. A sixth mass extinction is currently underway, driven by human activity. Habitat destruction, pollution, climate change, and overexploitation are driving species to extinction at rates far higher than the background extinction rate. Most paleontologists agree that the current extinction rate, often estimated at 100 to 1,000 times the background rate, exceeds all but the most severe natural mass extinctions. If current trends continue, perhaps 50% of species could be extinct within a few centuries. This would rival even the Permian extinction in severity. Unlike natural extinctions, this extinction is occurring rapidly and is driven by a single species, humans. This raises questions about whether life can recover as it has after previous mass extinctions. Recovery from natural extinctions takes millions of years. Can we prevent extinction rates from reaching that point? Additionally, humans have the unique ability to understand extinction risks and potentially change behavior to reduce them. Understanding how species have responded to past environmental changes provides insights into how modern species might respond to climate change and habitat loss. Species that survived past extinctions tended to be generalists with broad diet preferences, high reproductive rates, and broad geographic ranges. Species most vulnerable were specialists with narrow preferences, slow reproduction, and limited ranges. This suggests that modern conservation should prioritize protecting generalist species and expanding habitat corridors to allow species movement as climates change. It also suggests that reducing additional human stressors, pollution, overexploitation, might allow species to better tolerate climate change stress. Understanding evolution, the mechanism of life's diversity, natural selection, the engine of evolution. Evolution is perhaps the most important concept in biology, yet it's often misunderstood. Evolution is not a process with direction or purpose. It doesn't result in progress or improvement. Rather, it's a process of change over time, driven by the mechanisms of variation, heredity, and natural selection. Charles Darwin's insight, now over 150 years old, remains central. Organisms vary within populations. Some individuals are taller, faster, stronger, better camouflaged, or better fed than others. This variation is, at least partly, heritable. Offspring tend to resemble their parents. If variation in a trait is correlated with survival or reproductive success, then individuals with those favorable traits will leave more offspring. Over generations, the proportion of individuals with favorable traits increases in the population. This is natural selection. Natural selection is not conscious or purposeful. It's not an external force directing evolution towards some goal. It's simply a mathematical consequence of variation, heredity, and differential reproduction. Individuals with traits that enhance survival or reproduction will leave more offspring. Their offspring will inherit those beneficial traits. Therefore, those traits will become more common in the population over generations. This simple process, operating over millions of years, has produced the incredible diversity of life on Earth. Small changes, accumulated over vast stretches of time, result in large morphological changes. Species diverge into multiple species. Organisms become more or less specialized. Body plans change dramatically. But natural selection alone doesn't explain all evolution. Genetic drift, random changes in gene frequencies due to chance, also plays a role, particularly in small populations. Mutation provides the raw material for variation. Sexual recombination reshuffles existing genes into new combinations. Gene flow between populations introduces new genes into a population from elsewhere. All these mechanisms work together to produce evolution. In large populations, natural selection is the dominant force. In small populations, drift becomes more important. In different environments, different selective pressures operate. The result is the continuous change and diversification of life. The fossil record as evidence for evolution. The fossil record provides direct evidence for evolution. It shows species changing through time, demonstrates the origin of new traits, and reveals the branching pattern of evolutionary lineages. Consider the evolution of whales, one of the best documented evolutionary transitions. Modern whales are clearly mammals. They breathe air, nurse their young, have hair, albeit reduced in some species, and possess skeletal features indicating mammalian ancestry. Yet whales live in the ocean and have many features adapted to aquatic life: streamlined body, flippers, blowholes. How did whales evolve from terrestrial mammals? The fossil record shows the transition. Early whale ancestors like Ambulocetus, a 49-million-year-old transitional form, had characteristics of both land mammals and whales. It had a mammalian skull and teeth, but a body adapted for aquatic hunting. Its limbs were intermediate between terrestrial legs and aquatic flippers. Subsequent fossils show progressive modification. Later whales had more fully aquatic bodies, more modified forelimbs, modified hind limbs. Modern whales retain hind limb bones, albeit reduced and non-functional, and retain the tetrapod body plan modified for aquatic life. The fossil record documents this transition in detail. Each successive species shows incremental change, with whale ancestors gradually becoming more whale-like over millions of years. This is exactly what evolutionary theory predicts: gradual change over time, with each change providing an advantage in the organism's environment. Similar documentation exists for the evolution of birds from theropod dinosaurs. Archaeopteryx, a 150-million-year-old fossil, has features of both dinosaurs: teeth, clawed wings, long bony tail, and birds, feathers, wishbone, ability to fly. Subsequent fossils show birds becoming increasingly modern. Living birds represent the latest step in a 150-million-year evolutionary journey from theropod dinosaurs. The evolution of tetrapods from fish shows a similar pattern. Fish with lobed fins, like coelacanth and lungfish, show the closest affinity to tetrapods. Tiktaalik, a 375-million-year-old fossil, has features of both fish, scales, fins, gills, and tetrapods, neck, robust limbs, digits. Its discovery in 2004 filled a gap in our understanding of the fish-tetrapod transition, providing a prediction confirmation of evolutionary theory. These and many examples in the fossil record demonstrate evolution's reality. The fossil record is incomplete. Most organisms never fossilize, but it preserves enough information to document major evolutionary transitions. And every documented transition supports evolutionary theory: gradual change, modification of existing structures, conservation of fundamental body plans. Adaptive radiation and the origin of diversity following mass extinctions. We see adaptive radiations, periods of rapid speciation and morphological evolution. The classic example is the radiation of mammals in the Paleocene and Eocene epochs, following the KPG extinction that killed the dinosaurs. Before the extinction, mammals were small, unspecialized creatures. After the extinction, in the absence of dinosaurs, they diversified rapidly. Within 20 million years, major mammalian lineages had evolved: primates, carnivores, ungulates, hoofed mammals, rodents, whales, bats, and others. Each of these lineages went on to diversify further into multiple species. What allowed this rapid radiation? The extinction of dinosaurs removed the competitive dominance of those species. Ecological niches that dinosaurs had occupied became vacant. Mammals, with their adaptability and high reproductive rates, rapidly evolved to fill those niches. The environmental opportunity allowed rapid evolution. This pattern of rapid evolution in response to opportunity is seen repeatedly. When species are introduced to new environments with vacant niches, rapid evolution often occurs. Darwin's finches in the Galapagos Islands show rapid evolution in response to environmental change. Hawaiian honeycreepers show adaptive radiation in response to the availability of multiple ecological niches on the islands. Adaptive radiation demonstrates an important principle: evolution is not uniformly slow. It can be rapid when conditions permit. But it requires both time and opportunity. A species cannot evolve faster than its generation time allows. Large animals with long generation times evolve slowly. Small animals with short generation times can evolve more rapidly. Similarly, evolution accelerates when selective pressures are strong and when substantial genetic variation exists in the population. The molecular basis of evolution. While paleontology focuses on morphological change, molecular biology reveals the genetic basis of evolution. Comparing DNA sequences between species reveals how recently they shared a common ancestor. Species that diverged recently have similar DNA sequences. Species that diverged long ago have more different sequences. These molecular comparisons have revealed evolutionary relationships that weren't obvious from morphology alone. Humans and chimpanzees are about 98% identical in DNA sequence. Humans and dogs are about 85% identical. Humans and fruit flies are about 40% identical. These differences reflect the time that has passed since species last shared a common ancestor. Molecular analysis has also revealed the mechanisms of genetic change. Mutations, copying errors when DNA is replicated, are the source of new genetic variation. Most mutations are neutral, having no effect on survival or reproduction. Some mutations are harmful and are selected against. Rare mutations are beneficial and are selected for. Molecular analysis has revealed genes that control development, how organisms develop from a single fertilized egg into a complex adult. These developmental genes are remarkably similar between distantly related species. A fruit fly has genes controlling eye development that are nearly identical to human eye development genes. This suggests that these genes originated in a common ancestor and have been preserved because they perform essential functions. Molecular evolution is occurring in real time. Viruses like influenza and COVID-19 evolve rapidly, producing new strains as mutations accumulate. Bacteria evolve antibiotic resistance, sometimes within years of new antibiotics being introduced. These real-time observations of evolution demonstrate that the mechanisms paleontologists infer from the fossil record actually operate in living organisms. Adaptation and environmental response. How species adapt to environmental change. Life on Earth has always faced environmental challenges. Climate fluctuates. Ecosystems change. Competitors evolve. Predators and parasites co-evolve. Species that cannot adapt to environmental change go extinct. Species that can adapt persist and often thrive. Adaptation occurs through multiple mechanisms. Genetic change via natural selection is the primary mechanism on evolutionary timescales. But on shorter timescales, organisms can adapt through behavioral flexibility, physiological adjustment, and developmental plasticity. Behavioral flexibility allows organisms to adjust behavior to changing conditions. A predator that usually hunts at dawn might shift to hunting at dusk if prey become nocturnal. An herbivore might shift diet to different plants if preferred plants become unavailable. These behavioral adjustments allow organisms to exploit new resources without requiring genetic change. Physiological adjustment occurs within the lifetime of an organism. An organism exposed to cold can increase metabolic rate to maintain body temperature. An organism exposed to high altitude can increase red blood cell production to enhance oxygen carrying capacity. These adjustments allow organisms to tolerate varying environmental conditions. Developmental plasticity allows an organism's development to be influenced by environmental conditions, producing different phenotypes from the same genotype. A tadpole raised in predator-rich environments might develop larger mouth and broader tail than a tadpole raised in predator-free environments. These developmental adjustments allow organisms to adjust their investment in different traits based on environmental demands. Over longer timescales, genetic change permits more fundamental adaptations. Organisms that survive in a novel environment for many generations often show genetic changes, evolution of new traits through natural selection. For example, when humans introduced cane toads to Australia to control insects, the toad's predators evolved resistance to toad toxins. Snakes and other predators that could tolerate the toxins survived and reproduced more successfully. Within decades, populations of snakes had evolved higher toxin tolerance. Climate change and species response. Climate change is perhaps the most significant environmental challenge facing modern species. Changes in temperature, precipitation, and seasonality affect every organism. Paleontological records of past climate change provide insights into how modern species might respond. During the last glacial maximum, approximately 20,000 years ago, global temperatures were roughly 5 to 10 degrees Celsius colder than today. Ice sheets covered much of North America and Europe. Landscapes that are now temperate were covered in tundra or boreal forest. Sea levels were lower, with water locked in ice sheets. Species distributions shifted dramatically. The paleontological record shows how species responded. Some species migrated toward warmer regions. Some became extinct in areas where climate became inhospitable. Some evolved to tolerate colder conditions. Others went extinct entirely. When the glaciers melted and climate warmed, species shifted back. Species followed their preferred climates poleward and to higher altitudes. Boreal forests replaced tundra. Temperate forests replaced boreal forests. These shifts took thousands of years. Species couldn't evolve fast enough to adapt to rapid climate change. Instead, they either migrated or went extinct. This historical pattern suggests that modern rapid climate change will similarly stress species. Those able to migrate toward favorable climate might survive. Those unable to migrate might go extinct. However, modern climate change is occurring more rapidly than the climate changes species experienced after the last glacial maximum. Some species might not be able to migrate fast enough. Others might not find suitable habitat in regions where climate remains favorable due to human development, or other species having colonized those regions already. Biological invasions and ecosystem disruption. Species have always competed and interacted. But historically, geographic separation limited competition. Species in Europe interacted with European species. Species in Australia interacted with Australian species. Species in the Americas interacted with American species. This separation lasted until relatively recently, when humans began transporting species around the world, both intentionally and accidentally. When a species is introduced to a new region where it didn't previously occur, ecological disruption often results. The introduced species might be a predator with no natural enemies in the new region, or an herbivore with no herbivory defenses evolved in plants of the new region. Without natural enemies, introduced species sometimes proliferate, outcompeting native species and disrupting ecosystems. Examples are legion. Rabbits introduced to Australia exploded in population, competing with native species for vegetation. Rats introduced to islands have driven numerous species to extinction. Kudzu introduced to the American South has become an invasive vine that smothers native vegetation. Zebra mussels introduced to the Great Lakes compete with native mussels and clog water intake pipes. Invasive species demonstrate the importance of competitive dynamics and coevolution in ecology. Native species have evolved to compete with other native species and to defend against native predators and parasites. When an exotic species arrives, native species often lack defenses. The new competitive environment disrupts established ecological relationships. Interestingly, paleontological records show that invasive species have always affected ecosystems. When species have been able to disperse to new regions, via land bridges, for example, invasion and disruption have occurred. The extinctions of megafauna in Australia, North America, and other regions occurred after human arrival, itself a form of biological invasion. Humans, a successful species with tools and intelligence, invaded new continents and contributed to the extinction of naive megafauna. Astrobiology, life beyond Earth, the search for extraterrestrial life. If life is common on Earth, existing in nearly every conceivable environment, from boiling hot springs to frozen glaciers, could life exist elsewhere in the universe? This is the central question of astrobiology, the study of life in the cosmos. The universe is vast beyond comprehension. Our galaxy, the Milky Way, contains perhaps 200 to 400 billion stars. Most stars likely have orbiting planets. Current estimates suggest that there are more planets in the universe than stars, trillions upon trillions of them. If even a tiny fraction of these planets harbor life, life might be common in the universe. But detecting life on distant planets is extraordinarily challenging. No confirmed extraterrestrial life has been discovered. But the search continues, and the methods have become increasingly sophisticated. One approach is the study of exoplanet atmospheres. When a planet passes in front of its star from our perspective, a small amount of starlight passes through the planet's atmosphere. Analyzing this light reveals what gases are present in the atmosphere. If an atmosphere showed signs of biological gases, oxygen, methane, or other compounds that would quickly disappear without biological replenishment, it would suggest life. Another approach is direct imaging of exoplanets, followed by spectroscopic analysis to search for signs of life. A few exoplanets have been directly imaged, and analysis of their light might reveal biosignatures. A third approach is radio astronomy. Any technological civilization would likely broadcast radio signals. Searches for these signals, conducted by projects like SETI, search for extraterrestrial intelligence, listen for artificial signals from space. Habitable zones and the possibility of life for life as we understand it, liquid water is probably necessary. Organisms on Earth require water as a solvent for biochemistry. In the absence of water, chemistry operates differently, and complex chemistry essential for life might not occur. This observation has led to the concept of habitable zones, regions around stars where planetary temperatures allow liquid water to exist on the surface. Too close to the star, and planets are too hot for water. Too far, and planets are too cold. In the habitable zone, water can exist in liquid form. Earth is in the habitable zone of the Sun. Mars is at the edge of the habitable zone. Venus is too close, with temperatures too hot for liquid water. Jupiter and Saturn's moons are too far in the main habitable zone but might have liquid water under ice sheets due to tidal heating from their parent planets. The concept of habitable zones has been refined in recent years. Earth-like planets, rocky planets in the habitable zone of Sun-like stars, have become a focus of exoplanet searches. Missions like the Kepler Space Telescope have discovered thousands of exoplanets, including many in habitable zones of their stars. Some of these exoplanet discoveries are tantalizing. Proxima Centauri, the nearest star to our Sun, has an exoplanet in its habitable zone. TRAPPIST-1, a nearby star, has multiple planets in the habitable zone. If any of these planets have water and appropriate chemistry, they might harbor life. Extremophiles and the expanded habitable zone. The discovery of extremophiles on Earth has expanded our thinking about where life might exist elsewhere. If life can exist in boiling hot springs, frozen tundra, highly acidic environments, and the vacuum of space, then perhaps life could exist on worlds we previously thought inhospitable. Europa, a moon of Jupiter, has a surface of ice covering an ocean of liquid water. The ice is kept warm by tidal heating from Jupiter's gravity. The ocean is dark, beneath kilometers of ice, isolated from the sun's light. Yet extremophiles on Earth thrive in similar conditions, in deep ocean hydrothermal vents, where hot water and minerals from Earth's interior create an energy-rich environment despite the absence of sunlight. If Europa's ocean has hydrothermal vents, it might harbor extremophile microorganisms similar to those in Earth's ocean vents. Life might not require sunlight, or a habitable zone around a star. It might require only liquid water and an energy source. Enceladus, a moon of Saturn, similarly has an ocean of liquid water beneath an icy crust. Geysers on Enceladus have been detected, suggesting that liquid water reaches the surface. The water contains organic compounds, detected by the Cassini spacecraft. If Enceladus's ocean has hydrothermal vents and appropriate chemistry, it might harbor life. Titan, another moon of Saturn, has a surface of water ice and hydrocarbon lakes. Its atmosphere is thicker than Earth's, composed primarily of nitrogen with methane clouds. Scientists have long speculated about possible methane-based life on Titan, organisms that use methane instead of water as a solvent, and hydrogen instead of oxygen for respiration. Such organisms would be fundamentally different from Earth life, but the principles of chemistry and evolution might still apply. The Drake equation and the frequency of intelligent life. In 1961, astronomer Frank Drake proposed a method for estimating the number of technological civilizations in our galaxy. The Drake equation multiplies together factors including the rate of star formation, the fraction of stars with planets, the fraction of planets that could harbor life, and other factors, yielding an estimate of the number of intelligent civilizations. The problem is that most factors in the Drake equation are unknown. We can estimate the rate of star formation from astronomical observations. We know that many stars have planets. But we don't know what fraction of planets have conditions suitable for life. We don't know how often life arises. We don't know how often life evolves intelligence. We don't know how long civilizations persist. Estimates for the Drake equation vary wildly, from suggesting that intelligent life is extremely rare, perhaps just us in our galaxy, to suggesting that intelligent life is common, perhaps millions of civilizations in our galaxy. One useful observation is the Fermi paradox. If intelligent life is common, where is everyone? We see no evidence of extraterrestrial civilizations. No radio signals detected. No obvious megastructures observed. No evidence of interstellar travel or colonization. Why? Several hypotheses address the Fermi paradox. Perhaps intelligent life is indeed rare, and we might be alone in our galaxy. Perhaps intelligent life is common but short-lived, with civilizations destroying themselves before achieving interstellar travel. Perhaps space is so vast that even common intelligent life is spread so thinly that contact is unlikely. Perhaps intelligent life prefers to avoid contact with others. The Fermi paradox reminds us of how little we actually know about the possibility of extraterrestrial life. Absence of evidence is not evidence of absence. Just because we haven't detected extraterrestrial life doesn't mean it doesn't exist. The future of paleontology and astrobiology, new technologies on the horizon. Paleontology continues to advance as technology improves. Several emerging technologies promise to revolutionize the field further. Synchrotron radiation is a powerful form of X-ray radiation that can image fossils at extremely high resolution, revealing fine details of cellular structure. Synchrotron radiation has revealed internal structures of ancient insects preserved in amber, showing cells, organelles, and even DNA in some cases. Advanced mass spectrometry allows precise identification of chemical compounds in fossils. This technique has revealed the presence of original biochemistry in some fossils, proteins and other molecules preserved for millions of years. Understanding how molecules are preserved in fossils provides insights into the minimal conditions needed for life. Artificial intelligence is beginning to be applied to paleontological data. Machine learning algorithms can identify fossils, classify them, and predict missing information from incomplete specimens. As paleontological databases grow, AI will increasingly help paleontologists analyze large datasets and identify patterns humans might miss. Gene synthesis technology might eventually allow creation of ancient organisms from genetic information recovered from fossils. The first steps toward de-extinction of mammoths have already been made. Future technological advancement might allow resurrection of extinct species based on genetic information preserved in fossils. Future missions and searches for life. NASA and other space agencies are planning missions to search for life on other worlds. The James Webb Space Telescope, recently deployed, has the capability to analyze exoplanet atmospheres for biosignatures. Future missions to Europe and Enceladus will search for possible microbial life in subsurface oceans. A future mission to the deep ocean vents of Enceladus might confirm whether life exists there. If it does, it would suggest that life is common in the universe. If microbial life existed on two different moons, it would indicate that the origin of life is not extraordinarily improbable. Similarly, if microbial fossils are discovered in the subsurface of Mars, it would suggest that life can arise independently on different planets. Two independent origins of life would suggest that life is common wherever conditions permit. The philosophical implications of life's diversity, as we've explored throughout this documentary, life exhibits remarkable diversity, resilience, and creativity. From the Cambrian explosion to the bizarre dinosaurs to the extremophiles that defied biology, life has repeatedly demonstrated an ability to evolve in unexpected directions and survive under extreme conditions. This diversity suggests several things about life. First, life is fundamentally adaptable. Evolution is a powerful process that generates solutions to environmental challenges. Given time, organisms will evolve to exploit available resources and adapt to changing conditions. Second, life is resilient. Despite multiple mass extinctions, despite environmental catastrophes, life persists. Organisms that might seem fragile, a microscopic tardigrade or a small mammal, prove capable of surviving conditions that would kill seemingly robust organisms. Life finds a way. Third, life is creative. Evolution explores evolutionary space, trying different solutions to environmental problems. Some solutions prove successful. Others fail. But the diversity that results is remarkable, far beyond what anyone might have imagined prior to understanding evolution. These characteristics suggest that if life exists elsewhere in the universe, it might be similarly diverse and resilient. The specific organisms would be different, adapted to the particular conditions of their world. But the principles of variation, heredity, and natural selection should operate similarly. Life elsewhere might not be carbon-based or require water, but some form of chemistry capable of replication and evolution would likely apply. Conclusion: a universe full of possibility. As we conclude this exploration of paleontological discovery, evolutionary mechanisms, and astrobiological possibilities, we're left with a perspective on life that is humbling and inspirational. Humbling, because we recognize that humans are not the culmination of evolution. We are not the ultimate form toward which all previous life was building. We are simply the current dominant large animal on Earth, occupying a particular ecological role in a particular time. Evolution will continue, and if humans persist, we will continue to evolve. If we go extinct, life will continue on Earth, and other species will diversify to fill the ecological roles we occupied. Inspirational, because we recognize the incredible adaptability and resilience of life. Life has survived every challenge thrown at it by Earth. Life has responded to environmental change by adapting, by persisting. This resilience is a testament to the power of evolution and the fundamental robustness of living systems. The study of paleontology shows us a world transformed by time. Species that once dominated the Earth, dinosaurs, megafauna, countless smaller organisms, are now extinct, known to us only through fossils. Yet in their time, they were successful, they thrived, they evolved to fit their world. That success, and their eventual extinction, reminds us that the world is always changing, and nothing is permanent. The study of evolution shows us the mechanism through which life has responded to that change. Natural selection, working on variation within populations, has shaped organisms to fit their environments. Over millions of years, small changes have accumulated to produce the incredible diversity of life. The study of extremophiles shows us that life is more robust and creative than we might have imagined. Organisms exist in environments we thought impossible for life. They use biochemistry we didn't know was possible. They demonstrate that life's flexibility extends far beyond what we observe in typical environments. The search for life beyond Earth is the logical extension of these discoveries. If life is creative enough to exist in boiling hot springs and frozen deserts, if life is resilient enough to survive vacuum and radiation, then perhaps life is common in the universe, existing on worlds we haven't yet explored, under conditions we might not immediately recognize as conducive to life. The future of paleontology and astrobiology lies in continued exploration and discovery. New fossils are discovered each year. New exoplanets are detected. New understanding of extremophiles emerges. Each discovery adds to our knowledge of life's diversity and possibilities. And yet, despite all our discoveries and all our understanding, tremendous mysteries remain. We don't fully understand how life originated. We don't know whether life is common in the universe. We don't understand all the mechanisms of evolution and adaptation. We haven't discovered most species that have ever lived. The fossil record preserves only a tiny fraction of the organisms that existed. These unsolved mysteries are what drive science forward. They inspire scientists to ask new questions, to develop new technologies, to explore new frontiers. The mysteries of life, past, present, and potentially present on other worlds, represent the frontier of human knowledge. Final thoughts: we live on a living planet, a sphere of rock orbited by the sun, covered with ecosystems teeming with life. That life has a history stretching back nearly 4 billion years. That history is one of continuous change, adaptation, innovation, and evolution. The creatures we've discussed, the Irvingtonian megafauna, the coelacanth that returned from extinction's brink, the bizarre creatures of the Cambrian, the strange dinosaurs that walked ancient Earth, the extremophiles that defied biology, all are part of that history. They are part of the story of life on Earth. And we are part of that story as well. Humans are the latest chapter in the unfolding narrative of evolution. We possess intelligence, technology, and the ability to understand the history of life. This gives us unique responsibility and opportunity. We can choose to preserve the remaining species on Earth, to understand the processes that have shaped life, and to search for life beyond our world. The study of fossils and astrobiology reminds us that we are part of something larger than ourselves, a living universe, full of possibility, full of mystery, full of wonder. That perspective should inspire us to care for the life on our planet and to remain curious about the possibilities beyond it. Thank you for joining us on this journey through time and space, through the fossil record and the mechanisms of evolution, through the remarkable resilience of life in its many forms. We hope you've found the discoveries as fascinating as we have. If you've made it this far, please let us know in the comments what you thought of this documentary. What discoveries fascinated you most? What mysteries do you find most compelling? Your feedback helps us create content that resonates with viewers and explores topics that matter to you. Subscribe to the channel for future videos exploring the mysteries of our world, the history of life on Earth, and the possibilities of life beyond our planet. The story of life is still being written, both by evolution continuing on Earth and by our expanding search for life elsewhere in the cosmos. Until next time, keep looking up and down. The universe holds secrets waiting to be discovered, and the fossil record beneath our feet still has stories to tell. Thank you for watching this comprehensive exploration of paleontological discovery, evolutionary mechanisms, and astrobiological possibilities. Please remember to like, comment, and subscribe for more content exploring the wonders of natural history and science. See you in the next video. Welcome to a journey through time itself, where the earth beneath our feet holds secrets older than human civilization could ever comprehend. Imagine digging into the ground and discovering creatures that walked this planet millions of years ago, being so strange and wonderful that they challenge everything we thought we knew about life's possibilities. This is not fantasy. This is science. This is the real story of our planet's incredible past, and the resilience of life in ways that seem almost impossible. Throughout history, paleontologists and scientists have been nature's detectives, piecing together clues from fossils, ancient rocks, and the remains of creatures long extinct. Their discoveries have shown us that Earth has always been a place of wonder, danger, and continuous transformation. From the depths of ancient sinkholes to the edges of knowledge itself, we're about to explore the most fascinating discoveries that have reshaped our understanding of life, death, and everything between. This documentary examines some of the most incredible paleontological breakthroughs, mysterious creatures that seem pulled from mythology, the astonishing evolution of dinosaurs into forms we never imagined, and perhaps most remarkably, the microscopic organisms that can survive anything, literally anything, that the universe throws at them. So settle in, find a comfortable place to watch, and let us know in the comments where you're viewing this history video and what time you're watching it. Be sure to subscribe to the channel for future videos exploring the mysteries of our world. Now, let's begin our descent into the deep past. Part 1. The Fossil Record and Ancient Discoveries. The foundation of everything we know about prehistoric life rests upon a single, profound concept: fossils are messages from the past. They are nature's way of leaving behind a record of what once was. But understanding these messages requires patience, expertise, and sometimes, remarkable luck. The Florida's sinkhole, gateway to a forgotten world in the state of Florida, beneath the modern world of highways and cities, lies something extraordinary. Deep underground, natural sinkholes have created perfect preserves for ancient life. These sinkholes form when limestone bedrock dissolves beneath the surface, creating cavities that eventually collapse, leaving behind depression-like formations. But what makes Florida's sinkholes so special is their ability to preserve organic material in ways that normal fossil sites simply cannot. Around 1251 in our timeline of discoveries, paleontologists made remarkable finds within one of these sinkholes. The creatures found here came from the Irvingtonian period, a geological age that has long remained mysterious to science. The Irvingtonian period, spanning roughly from 300,000 to 100,000 years ago, represents a transitional time in